Okay, so I have 30 minutes to tell you guys how to do image analysis, which is obviously an impossible task. So this will be a ridiculously broad overview. And while thinking about giving this talk, the first and most obvious question that I had to deal with is whether or not I was going to try to talk about image analysis to someone who knows how to program and someone who doesn't know how to program. And I think, you know, this is a crisis that I think a number of biologists go through at some point in their career. And I think the answer, unlike Hamlet, is really clear. You should learn how to program. You should do it sooner rather than later. You're just going to get more busy. Your brain is going to get less plastic. You're going to be better off. You're wasting years of your life clicking and doing things. But that is a bit, albeit a biased perspective, but I think that's really true. That being said, I don't want to speak only to people who know how to program. So this whole presentation has been aimed for someone who doesn't know how to program. And I hope that there are also things that people who know how to program will find valuable. So the outline.
Where's this background from?
This background is a fake bow, where you take the Z location of the pixel and then you encode it in color, so it looks very pretty. Yeah.
Is that an ImageJ plugin?
I'm not sure. I think it's an Amaris plugin in this case. Yeah. So the outline of the talk is, first I'm just going to talk about some very general principles about image analysis, which I think kind of speak to programmers or non-programmers alike. Then I'm going to talk, as Alex said, about some various open source packages that exist to help you with your image analysis. And then finally, I'm going to talk about some common artifacts that come up in the course of imaging and some things that you can do to try to deal with them in the analysis step. So my first piece of general advice is that analysis is really, even though it comes at the end of your experiment, it really isn't the last step. You don't want to be a scientist that sort of feels like this, where they have a giant pile of data that they haven't looked at yet and they're trying to get some result out of. The reason for this is not just so you don't feel so overwhelmed, as she probably does, but that it's not obvious necessarily what aspects of the experiment are really the most important in order to make your data analyzable. And so if I would strongly urge you, once you got that experiment working, to take the week off and try to analyze some data in order to really understand how you're going to analyze the data, what are the critical aspects of the data that make it easier or harder to analyze, and give you a realistic expectation about how long it's going to take you to analyze your data. So it may seem like that week is a bit of a waste of time when you think you got everything working, but it's sure of a hell less of a waste of time than having to redo months and months of experiments, not to mention less expensive. The second piece of advice, in thinking about any image analysis pipeline, there are going to be parts of the pipeline which are subjective, ranging from something as simple as where do I set this threshold in order to extract cells from this picture, to something much more obvious like method sections that read images were scored by a human expert. The best way to convince yourself and to convince other people that you haven't cheated or subconsciously biased yourself during this analysis is to blind yourself. Not only does this the rigorous way to do things, it also really helps you and forces you to structure your data so you don't end up with some kind of disorganized mess after the place, because you need to create a file which has a list of all of the data that you've got and what condition it came from, and come up for a way to produce randomized file names, and that in itself can serve as the template for your analysis of the different conditions after the fact. Finally, the kind of flip side to a blinded analysis is that it's also really important just to look at your data, get really, really intimate with it and stare at it. And to serve as an example of that, one of the favorite stories of mine I heard from the guy whose last name I have trouble remembering. No, actually, his competitor.
Oh, his enemy.
Not Takahashi, it's different than that. Guy from Texas.
Yeah.
The last author on this paper here. So both Mignot and him sort of came into the same field, and his version of the story is that the scientist wanted to do something useful, and so he thought, you know, most of our drugs are G-protein, affect G-protein coupled receptors, and there's a bunch of orphan G-protein coupled receptors that we don't know their ligands for, and so They went through the brain, screening for, they took the whole brain and made whole brain lysates and took each little fraction and tried to find the fraction that caused the receptor to light up, and they found one. They were excited, and they fed this ligand that they purified to rats, and they thought that seemed to affect their eating behavior, and so that was the first paper. But then they made the knockout mouse, and they really didn't see anything wrong with the knockout mouse, or at least that's how the story goes, until they started filming the mice at night. And at least as the PI of the story, the PI of this paper says, really it was a student who was watching this, and you saw the mouse run around, and then all of a sudden it stopped. And it wasn't until they really just watched these overnight movies did they notice this happening. And you can imagine that if you had a really fancy automated system that you just plugged these movies into, and it tracked locations of all the mice and gave you their average time spent running or not running, that the effect of this narcoleptic mouse might be really subtle. But it's really the fact that someone spent the time to really know what is a natural kind of way a mouse falls asleep, and one that's sort of aberrant like this. Eventually this mouse is going to get back up again, like that. And back to the previous point, if you read the methods section of this paper, it says open field narcoleptic episodes were scored with observers blinded to genotype for 9 homozygous and knockout mice, right? So it really is, they didn't have any fancy analysis, they just had, that they saw that there was something weird. Okay, so that's it for general advice. Onto free resources. So I'm going to talk about 3 different programs that are available. The first is Fiji. Fiji is a very clever acronym which stands for Fiji is just ImageJ, and that's what it is. It is just ImageJ, which is a project run from the NIH, but it just has a whole ton of extra plugins added on. So this is just example 1 submenu of the plugins folder within Fiji, which has an ungodly number of things. So I'm obviously not going to tell you about all the things Fiji can do. The best way to find out if Fiji can do something is type Fiji ImageJ and then some description about what it is that you want to do, and it probably has it somewhere. The great things about Fiji is it's a completely open source project. It's also, they make it really easy to make macros, which are kind of like programming for beginners. You click on a bunch of things, and then it writes down what is it that you did, and then you can just hit play, and it will redo exactly those set of things. But for more serious programmers, it's scriptable. You can extend it in Jython, in Bean Shell, in Java. The Smith Lab uses Fiji for all of our image analysis pipelines, and we write lots and lots of code that branch off of Fiji. Another program very much in the same vein as Fiji is called ICE. Its website is here. ICE is also a GUI interface, just like Fiji. It has lots of plugins. It's also scriptable with JavaScript or Python, and it also interfaces really well with MATLAB, as well as ImageJ. I mention it here mostly because I'm really impressed with its ability to do tracking and to find puncta in images, and to find puncta in cells in images, and to track them over time. So here's an example of it just using a fancy wavelet algorithm to pick out noisy little puncta in a movie. It does a pretty good job of that. Maybe even more impressive is this example they call active cells. So it's this plugin here which kind of is able to really smartly find the outlines of cells within kind of noisy images. So he's going to kind of roughly say, okay, there's a cell there, and then play, ooh, and it zooms and finds the edge of it really well. And then he's going to show you how you can copy that, just kind of roughly place it over some other locations, and then let's see, we'll do 2 more, and then he's going to hit this double play button over here, and whoop, and it all magically finds the edge. It's really pretty impressive stuff. The final thing I'm going to talk about is Cell Profiler. Cell Profiler is, again, another open source Python-based program. And the great thing about Cell Profiler is it's really set up from its foundation to be an image pipeline. So it's the kind of thing that it's not do this thing once, it's rather set up so that you set up a set of things that it's going to do to any image or all the images in a folder, and then it will automatically, if you get it working for one, you can then apply it to all the rest of your data. And so it works by stringing together these sort of modules which have well-defined inputs and outputs. And there's a whole set of modules that are available. And maybe most importantly, one of the modules is run ImageJ. So any command that exists in ImageJ can then become a module which you can string together into Cell Profiler. Okay, questions, yeah?
Actually, is there, like I've used Cell Profiler in the past for doing something different than my current project, but one of the problems was I thought there was no way of following a given cell. So it was good for crossing through like stack of 2D images. But if you wanted to follow one cell in time or multiple cells with an image in time, it was actually fairly hard to do so. Do you know if that's...
I'm not an expert in Cell Profiler. I just know, I know it's, I like it. I think that's probably is still true, although I'm not an expert, so I wouldn't say, and I'm not sure whether or not I'm a Fiji expert and not an ImageJ expert. And it's one of the annoying things about Fiji is that it doesn't necessarily, it's not going to play well with Cell Profiler, at least right off the bat. But I don't know the answer to that. I can recommend for tracking purposes, I think the algorithms that are built into IC are really good. They have this sort of nice probabilistic tracker. You know, in tracking, you first need to segment objects in all the frames, and then you need to link together the objects across frames. And it uses pretty intelligent algorithms for doing that. And that, especially in that linking process, I think is a hard thing to do. And they've, I think, have an algorithm that works that is state of the art.
So Forrest, do you end up using some programs for, let's say, generating some aspect of the image and then might export it out in a format that then, say, Fiji might do a better job of doing? Do you do a lot of sort of cross-platform stuff?
In fact, I think this movie, for example, I don't know. IC isn't as good as I would like about exporting movies, and so and being able to play with different options. So I did this particle tracking here in IC, and then it just saved out for me a sort of mask where each of these little objects was white and everything was black. And then I converted it into these red outlines in Fiji and saved out an AVI in Fiji so I could show it to you as a movie, because I couldn't figure out how to get IC to show me a movie in the way that I wanted. And in general, yeah.
You kludge it together, kind of.
I kludge it together, and that's, you know, you have to, yeah.
Yeah.
Okay. Any other questions about open source stuff? Again, I couldn't possibly cover all the things that they can do. You're going to have to just look at them for what you specifically want. But I thought I would try to touch on some of the artifacts which are more universal in imaging, and there are problems, more general problems for people's analysis. So the first one is the one that we've talked about before, is bleaching. I got this movie from Emily. Is Emily? I've never met Emily. Okay, this is Emily. Okay, so Emily sent me this movie because she's trying to analyze, I think that, well, Emily can tell you, these are cells of some kind.
Yeah.
You want to say a little bit about what this is?
Would you rather? This movie is actually hers.
Oh, okay.
It's a secret project.
Yeah.
These are yeast cells, and so the function that you see is a protein that gets misfolded upon heat stress. So it will form these inclusions. And what we try to do is to define. So these are just wild type cells, and we are studying chaperones and protein folding systems. So we try to see how different chaperones affect, like, the occurrence of these puncta, the spatial localization, the coalescence of these puncta. So we count every puncta in the cell, and
right. So they'd love to, you know, count each of these little puncta, and of course the first thing you're going to do and have a problem with is the fact that the puncta are getting dimmer, and so you try a universal threshold that obviously is not going to work very well. So one thing that you might want to do in Fiji is run a bleaching correction, which essentially tries to find the rate at which the floor is bleaching and then normalize for that. So you can run this plug in here, adjust bleach correction, and there's different methods for doing it. I think the more principled one is the exponential fit, which just looks at the overall fluorescence of the image over time and then fits it to an exponential. And you can see, other than these few weird frames where something bizarre happened, it's actually a remarkably good fit to the data. And so then you just use this to normalize the overall intensity, and the result is this, a movie which basically doesn't look like it's bleaching. Now, it is still bleaching, but in this case there are so many photons being collected that you don't notice the relative change in the noise that much. You may notice that the background is not bleaching, is getting a little bit brighter as you go on, and that's because the out of the bleaching rate of the out-of-focus photons are not as high as the in-focus photons. Fluors, not photons. Okay, so once you fix the bleaching artifact, the next thing becomes obvious, which is, in fact, my favorite subject, which is motion artifact. The cells are sort of jiggling around a little bit. And again, Fiji has a plethora of plugins to deal with this. They're all in the well, most of them are in the registration part of the plugins folder. And I think that one of the best ones here is this descriptor-based series registration. And I could literally give a lecture for probably 45 minutes about how this algorithm works and why it works really well compared to other approaches. I don't really have time to do that, but the plugin has this nice interactive feature in which you can set most of the most important parameters interactively. And essentially, it's what the algorithm does is it tries to find sort of salient features of the image, and say this cell comes up as a local maximum once you run it through a kind of filter, and it's going to try to find these cells are arranged in a particular pattern, and it's going to try to find that pattern in each of the different sections, and then fit a transformation to put those patterns to overlap on top of each other. And the intelligence of the algorithm is it does that in a really smart way using an algorithm called Ransack that's really robust to lots of problems. Let's see. So once you set this up, you can see I can adjust the size of this to kind of make the size of the sigma be about the size of the cell, and that's how I get, it's kind of, you can see it's kind of detecting where the cells are basically. And you can side this threshold up and down to set it so it's selecting the best about the right cells. The bottom line is after you're done and you run it, you get a movie out like this, which has basically no shaking other than the shaking of the actual cells themselves, as well as the edges, because of course when you correct and slide the movie back and forth, the edge of the frame moves.
And Forrest, was the dots that you were showing before in the previous image from you were showing what Icy was doing, the movie that you got out, where you had the little red contours drawn?
Yeah.
Was that from this image as well?
Yes.
From this movie, okay.
This is from this. This is the first frame of this movie, which, so not only is there...
I'm actually talking about like 5 slides ago.
Yeah.
Oh, okay.
Right, so it was zoomed in,
yeah.
It was like one of these, it was like this area right here. I just wanted to show the puncta bigger, so I showed a smaller portion of it. Okay. So that takes care of most of this motion, except for if you pay really close attention. What you can see here at the beginning of the movie is that all the cells look kind of like blurred, and then slowly they come, the puncta become sort of more more punctate. I think that's because not only as their XY motion, but there's also Z motion through the movie, and that's probably because, I'm guessing now about your experiment, that you maybe have a heated plate or something.
It's a heated chamber. It's
a heated chamber, but when you initially had the chamber not heated, and then you put the chamber onto the heated, you had the cells off the heated chamber, and then you put them on the heated chamber. And so what's happening here is that as you collect the movie, the whole system is sort of reaching thermal equilibrium, and that's causing there to be these sort of slower drifts in the stability of the system. So this is an example of, well, okay, I could try to correct for this, but not really, right? I mean, basically the data is getting taken from the wrong focal plane. And the better way to deal with this kind of problem in the data, if you really decided that you cared about those first few frames where it was out of focus, is to do a better experiment, is to try to figure out a way to get the thing in thermal equilibrium before you start taking the movie. I didn't mean that as mean as it sounded.
Nobody likes it when the analysis guy starts coming in all big and telling the experimentalists what to do for us.
I know.
No, but the point you would say is to
warm
it up first.
Back to my original point about closing the loop between the analysis and the experiment is that if it might not seem like a big deal, but maybe it is a big deal, but you didn't really think about it unless you really started doing that analysis much sooner, and that would, yeah.
Mm-hmm. I actually, so like a lot of that though is actually something we can't possibly avoid, because if we were to warm it up, then these puncta would form and we would miss their initial formation. So a lot of this is just sort of figuring out what you can accept as errors in your data as well, right? So actually in the first few frames of that, there shouldn't have been any puncta for you to focus in on.
Really?
So
you maybe could try to heat the Yeah. No. Yeah.
Do a little
What-
autofocus.
Well, the other thing you can do is use these kind of real-time autofocus systems. I mean, it's
It
more
has
expensive.
that on there. It's a definite focus system. So the definite focus is engaged on that.
So that's the biology then of it being hazy.
Yeah,
and
so
then
there's no puncta at the beginning, and then as it warms up, the puncta form because it's a
So there's no-
temperature-sensitive mutant.
So they just diffuse.
There's no
if you
Z
just
drift.
keep it-
That's not. That's just all like going from diffuse to punctate.
Yeah.
Oh, okay. So I was confused.
Yeah, so usually we start like we do like the measurements like this is like a 30-minute movie, so
Gotcha.
like after at time point 0 when everything is diffused,
Oh.
and then the first
Okay.
dilation after 5 minutes or 4 or
The biology confused me into thinking that it was out of focus, but it's not.
Shows what you know now.
Yeah, I know. Yeah, exactly.
Coming in all big with your algorithms.
So I couldn't, because motion artifact is my favorite topic, I couldn't help but mention the fact that motion artifact in 2-photon microscopy is a very different sort of thing than in 1-photon, because it's a laser scanning technique, and the frame is not acquired all in a single snapshot, but rather you scan the beam back and forth. And so, because it takes you some amount of time for you to scan back and forth, when the animal moves, in this case this is a movie of one of those mice running on a ball of some cells which are just expressing YFP, they're not GCAM, in layer 2-3 of cortex, you can see that the frame kind of looks like it's like the brain is like all wobbly. That's not what's going on. Actually, the brain is moving relatively rigidly, but it's because the brain is moving a significant difference in the middle of the frame. And there are ways that you can deal with this. I spent a great deal of my PhD working on one, and you can read about it in this neuron paper. And there's also this other paper that has another method that is essentially solving the problem in the same way, in that you have to align each subpart of the frame independently. And on the left-hand side is the result of the motion-corrected movie, and you can see it basically looks static, except for these blank lines, which are an unavoidable problem because the beam is no longer passing over that portion of the sample, so you don't have any data. Okay.
Question. So,
Yeah.
I think this is a solution for the moving of the X and Y axis. So what about the Z?
Yeah. So the Z problem, you could try to, you could take a similar approach to try to predict. Essentially what you're doing is saying, okay, where did this line of data actually come from? And let's put it back where it belongs. If you were going to do the same thing in Z, you could do that. But if you acquired the data from the wrong frame, if this cell body is no longer within the frame, then you're out of luck, right? And so kind of the in-plane motion is a much better situation, because if I move the sample up, I'm still going to get to this spot later on in the frame, except for at the edges, right? But when you have the out of if you're not scanning in Z, then when you move in Z, you have, rather than blank lines, you've got blank slices, which is much worse.
So that might be a situation where you're going to try and tweak your experimental acquisition somehow to either get a smaller frame size or
to improve the way in which you do the surgery. So most of the tricks about getting this to work some are the ball, but the other is that Dan Dombeck really developed a lot of sort of better surgical techniques to hold the brain more constant as the mouse was running. Similarly, in the fly, people are doing this. They have these tricks where they make this like little presser foot that's got a hole cut out in it, and it like presses down on the fly brain to hold it still, and then the image through the hole in the presser foot. So there's lots of experimental tricks to try to reduce the motion. Dan and I used the joke that he tried very hard to make what I was doing irrelevant. Okay. Another very common problem is uneven illumination. This is a turf microscopy movie of microtubules growing on a cover slip. And turf is great, but it's really hard to get a very uniform illumination in turf. So this is kind of common to see. And so if you're trying to do some analysis which picks out where these microtubules are, it's kind of hard when you've got this kind of uneven illumination. You can do a kind of local contrast enhancement using, say, the subtract background tool in Fiji, which uses this rolling ball to look at what the local average is and then subtract that away from the image. And when you do that to this movie, you get something like this, which makes, you know, these microtubules up here stand out above the background closer than they are down here. Now, it's not perfect, obviously, but it's better than it was. The final problem, can we dim the lights for this? Probably.
Should I light some candles?
Yeah, okay. So is bleed through. So this is where you are requiring a multi-color image. And it may be, this is a 647 image and a 594 image of exactly the same area. And you might not see it in this image, but if I enhance the contrast, maybe you can notice some locations, say here, here, and here, in which the co-localization of the 647 and the 594 looks creepily equal. This is not because there's some amazing co-localization between these two molecules. This happens to be a stain for GABA. This is a stain for PSD95. Those are actually mostly exclusive, and this should be inhibitory neurons. This should be on excitatory synapses. What's going on is that the GABA stain is incredibly bright. And so you're using a really short exposure time here, and actually a longer exposure time here. And so you're getting some number of photons that you are actually getting exciting the 647 floor and being detected in the 594 channel. The way to deal with this is that what you need to do is to subtract a sort of percentage. You need to take this image and subtract it from this image with some factor. And you can figure out what that factor is just by looking at the plot. If each of these dots is a pixel, and the x-axis here is the intensity of the 647 channel, and the y-axis the intensity of the 594 channel, and the ratio of sort of how much light you're collecting in the 647 channel that's bleeding through into the 594 is set by the slope of this lower bound here. And so if you subtract this image times that slope from this image, you get this. So it basically 100% subtracts out that the GABA component from this PSD95 image.
So how can you do it? I mean, from ImageJ or you have to code it?
So I don't have a plugin for that. What I have is 20 lines of code. So this is just a little nugget to encourage you to learn how to program. It's really quite simple. I didn't use any fancy fitting techniques. I literally, drew a line and measured its slope like you did in high school, math class, and then, wrote down what the slope was and, subtracted the image times the slope. It's not rocket science. You just got to learn a little bit of syntax. So I guess that's all. I have an extra little bit about making a plugin that I was not necessarily going to do if we're over time.
Let's see, what time do we have? We're at 30 minutes now, so depending on how much time.
It's not going to take that long.
Okay.
It'll take 5 to 10 minutes. Okay, so I will come back to this. So I thought one thing that might be useful is to teach people how they can turn this kind of, click on this, do these sets of things to images into a pipeline in Fiji that will do this on a gazillion images, right? So luckily, Fiji has got a really nice thing to help you do this, to get you started. You can type in, if you click on here on plugins, on macros, and then record macro. When you start doing things, like if you did the set of things that I were talking to you about before, you'll get a little window which will produce this code for you. You don't have to type any of this, it'll just pop up. And if you hit play again, it would automatically do those 3 things. In this case, run the bleach correction, then the subtract background, and then run this registration tool. With the one caveat in that, rather than, if you remember, there was this interactive feature that let me slide the things back and forth. I don't want to do that if I'm going to process a bunch of images. I want to have it actually do something specific. So if we change that to advanced, we can actually type in numbers, and those numbers will come in over here in these things. And again, you don't actually have to do any coding. It just automatically writes this stuff down. Okay, so there's one other problem though, and that's that if you notice closely in this code, there's this portion here which is related to the file name that we're using. And we want to write something that's completely general. We want to have it, okay, I got my image open, and now I want to run this code, and I want it to do this set of things. So I need to know, I don't want to hard code this, I want it to be the title of the TIFF. So luckily there's a function in ImageJ which would tell you what the title is of the image that you've got open. So you call that function, get this variable title, and now I just want to replace this little bit with the title. So that's literally all you do. You change some code here to insert the title into this command. And now I've got this general script. I can hit run and it will do these three things in order. Okay, so the next thing that you would want to do is say, run that script on every image in a folder. So if you go to this website, you'll see that there's this nice little template, which is a code which will do whatever you say here on every file in the directory that you choose, and then save the results as a TIFF in another directory you choose. You don't need to write the code. You can just go to this website, copy it, and then basically paste in your macro right there. And so that's what you can do. So this is exactly the same macro that I showed you before, just pasted right there into that for loop. And now it's colored because I put it into the macro editor in Fiji. You run this and it will bleach correct and motion correct all of your data all at once. So it's ready to do the next step of the analysis. All right, so with that, I'm going to go back to my acknowledgements. Just wanted to say, as a, my acknowledgements are kind of framed here as a fake method section, in that you should do it as you can. Do cite Fiji, do cite people that write analysis papers like these people. They do a lot of work to write code for you, and it's good for their career. And yeah, thanks to Emily, and I'm sorry, your name?
Liliana.
Liliana?
Yeah.
Okay. For the live cell movies that I used in the demonstration, and my girlfriend Melissa, who gave me the tubular turf images.